presentation.
बाद में डिलीट कर दो बट कॉपी कर सकते हो।
हेलो, हाँ राहुल, उसको भेजना है, वो पेन ड्राइव ले जाएगा। वो ये
Anyway, it will be recorded. So
रिकॉर्डिंग में पेन ड्राइव लेके जाएंगे, पेन ड्राइव क्लास में।
yes.
बस रहने दे। बूटे रहने दे। होगी बेटा, पेन ड्राइव होगी। वहाँ पे लिखो चाहे वो ये वाला सामान है, ये वाली बीड़ी का एक लगा
Warm greetings and heartfelt wishes for a joyous day. It is my pleasure representing Dr. SSPUICT, Punjab University, to extend a warm welcome to each one of you as we come together to celebrate the 130th birth anniversary of esteemed Dr. Shanti Swarup Bhatnagar ji. This program has been organized in association with Society for Promotion of Science and Technology in India, SPSTI, Chandigarh chapters of National Academy of Sciences, India, NASI, INYAS, Indian National Youth Academy of Science, Institutions Innovation Council, Ministry of HRD India Initiative, and CRICS, Chandigarh Region Innovation and Knowledge Cluster. Before proceeding with the program, let us take a moment to explore the remarkable legacy of the revered Dr. Shanti Swarup Bhatnagar through an audiovisual presentation crafted by alumni of Dr. SSPUICT. I now invite our dynamic and vibrant chairperson, Professor Anupama Sharma, to present the welcome address. Before that, we would like to give a brief introduction of ma'am. Professor Anupama Sharma serves as the chairperson of Dr. SSBU ICT, Director of Energy Research Center, and is the former alumni relations at Punjab University, Chandigarh. Some notable academic contributions include: Ma'am is a member of Aspire Engineering Sciences Research Committee, Council for Scientific and Industrial Research. Ma'am is also a member of peer team for NAC and NBA accreditations. She has been bestowed with many noteworthy awards and recognitions. To mention a few: ACCTI, ACCT1 ETCR Award for her outstanding contribution published in Trends in Carbohydrate Research, Young Researcher Award, Asian Polymer Association in 2017, Career Award for Young Teachers, AICTE, India in the year 2006. She has a multitude of acclaimed papers and reputable books to her name. Professor Anupama Sharma, ma'am, please.
Before I give my welcome address, I request all the esteemed dignitaries to please occupy seats on the dais. Thank you, sir. A very good morning to all. Esteemed Professor Arun Grover ji, Professor Girish Sahani ji, Shri Sanjay Arora ji, Dr. Ashwani Kumar ji, Dr. Mohsen Rahmani ji, faculty and researchers from TRIC institutions, my colleagues from Punjab University departments, and dear students. A warm welcome to this Bhatnagar Day Oration. As we gather here today, we not only celebrate the memory of Dr. Bhatnagar, but also pay tribute to his unparalleled contributions to the field of science and technology. His pioneering efforts have left an indelible mark on the scientific landscape of our nation, and it is only befitting that we take a moment to reflect upon his vision and dedication. Dr. Shanti Swarup Bhatnagar, a luminary in the world of science, was not just a scientist, but a guiding force, a mentor, and a true visionary. His unwavering commitment to advancing scientific knowledge laid the foundation of Council of Scientific and Industrial Research at CSIR, University Grant Commission, and by extension, our very own Chemical Engineering Department, which was established in 1925 as Industrial Chemical Laboratory in Lahore. The Bhatnagar Day Oration serves as a reminder of our responsibility to carry forward Dr. Bhatnagar's legacy by fostering innovation, excellence, and collaboration in the field of chemical engineering. It is my profound pleasure to extend a warm welcome to stalwarts amongst us whose achievements and contributions have significantly shaped the realms of science, technology, and academia in India and abroad. Revered Professor Arun Grover ji, our mentor, who during his tenure as Vice Chancellor, Punjab University, initiated the celebrations of Bhatnagar Day to commemorate his pivotal contributions and also ensued award of TECHIC program to chemical engineering, as well as conceptualized and fructified the CRIC. He is true institution builder and has a knack of linking our past with our future. We are blessed with his presence to preside over the celebrations. We are honored to welcome Professor Girish Sahani ji, highly accomplished technocrat and former Director General of CSIR and INTEC. Dr. Sahani's leadership and vision have played a pivotal role in advancing scientific research and development in our country. His presence today amongst us is a testimony to the enduring commitment to excellence that he has exemplified throughout his illustrious career. We are privileged to welcome our very own Mr. Sanjay Arora ji, General Manager Marketing at GAIL. Our own alumnus 1987 batch, Shri Arora ji whose expertise in the industry and his dedication to innovation have contributed immensely to the growth and success of this field. We are delighted to have you here, sir, and we look forward to gain valuable insights from your experience. A warm welcome to Dr. Ashwini Kumarji, a distinguished Bhatnagar awardee. Dr. Kumar's outstanding contributions in his field not only earned him recognition, but have also inspired many in the scientific community. We are honored by your presence, sir. Another highly acclaimed speaker, Dr. Mohsen Rahmani, is a leader of advanced optics and photonics lab from our collaborating institution, that is Nottingham Trent University. Professor Rahmani's expertise and accomplishments in academia have made him ideally suitable to deliver today's expository talk and provide international orientation to this oration lecture. We also welcome distinguished faculty and researchers from our partnering institutions of Chandigarh Region Innovation and Knowledge Cluster, CRIC, as well as teaching departments of Punjab University. I acknowledge the presence of Dr. Professor Kiya Dharamveji and our faculty members whose responsibility is to carry forward the legacy of Dr. Bhatnagar. Our department, born out of Bhatnagar's vision, has grown and evolved over the years thanks to this relentless efforts of our faculty, researchers, students, and staff. We continue to strive for excellence, pushing the boundaries of knowledge and contributing to the progress of our nation. As we embark on this journey of enlightenment, let us remember and honor the legacy of Dr. Shanti Swarup Bhatnagar. May our interactions today foster collaborations, inspire innovation, and contribute to the continued advancement of science and technology. Thank you, and let's embrace the spirit of Bhatnagar Day with enthusiasm and dedication. Thank you so much.
Thank you, ma'am. I now request ma'am to present a floral welcome to our guests of honor, Dr. Girish Sahani ji. Shri Sanjay Arora ji, a DCT alumni of 1987 batch. Dr. Ashwini Kumar ji, recipient of 2022 Bhatnagar Award. And Professor Mohsen Rahmani from Nottingham Trent University, UK. Thank you, sir. So we are grateful for the venerable presence of Professor Arun Kumar Groverji, the former Vice Chancellor of Punjab University, Chandigarh. It is worth noting, as ma'am has said, that sir played a pivotal role in conceptualizing and inaugurating the series of Bhatnagar Day Oration lectures during his tenure. I would now request Professor Grover to share a few words on this occasion.
Good morning, all. So let me just at the outset clarify that the thought that we should have an annual oration in honor of Shanti Swarup Bhatnagar was mooted by eminent historian Professor Ramila Thapar, who was invited to deliver the PU Foundation Day lecture in 2012. We were at that time preparing to commemorate 150th birth year of Ruchiram Sahni, who was a mentor of Shanti Swarup Bhatnagar. When all these things were shared with her, which she did not know, she said everyone in the university must know these things. So please institute an annual lecture so that the entire university will be reminded every year of its own heritage by remembering not only Ruchiram Sahni but all others who got mentored as the science got permeated in Punjab. So Birbal Sani, son of Ruchiram Sani, Tantrisru Bhatnagar were the first botanists and chemists from India to be elected FRS. So the long story continues. So this is the background of this annual oration. And before concluding, let me just share that I was in BHU for a week till Sunday, and there I became aware that the BHU also holds an annual oration in the name of Shanti Swarup Bhatnagar on his birthday, that is today, February 21. So this year's speaker there is Professor Chandrasekhar, who was the former secretary of DST, and they have asked me to deliver the 2025 Bhatnagar oration at BHU next year on this day. So we are not the only institution which commemorates this titan of science in India. There are others who hold him in equally high regard. So I'm very happy that this tradition is continuing, and the chemical engineering department, FRIXI, and other institutions in the city are participating in this event. And we are also very, very happy that Professor Girish Sani, who is the Bhatnagar Fellow, after Shanti Swarup Bhatnagar, who is the next one from Punjab, who reached up to the coveted status of DG, CSIR, and after I think many years, we have a Bhatnagar awardee from the city, Dr. Ashwini Kumar. CSIR MTech has had Bhatnagar awardees in the past, but after a long back gap, we have Ashwini getting this award last year, and we have a distinguished alumnus of chemical engineering as part of this event, and NTU, our partner for about 10 years now. We visited Nottingham in 2014. So University of Nottingham and Nottingham Trent University, the two well-known universities from Nottingham on the international scene. TU has collaboration with both of them, and with NTU the interaction is very regular. There have been many visits, many collaboration amongst many departments, and I am very, very personally very happy that we have a speaker from NTU as well as part of today's event. Thank you very much.
Thank you, sir. Your words have always served as a source of inspiration, encouraging us to strive for excellence and persevere in our endeavors. Expressing deep gratitude for your enlightening and uplifting discourse, I request Chairperson Ma'am to felicitate Professor Arun Groverji. Thank you, ma'am. Thank you, sir. Continuing with the program, I am honored to introduce our distinguished guest of honor, Dr. Girish Saniji. Dr. Girish Sani is a biopharma inventor and former Director General, CSIR, and Secretary Government of India. Earlier, for about 10 years, he was the director of the Institute of Microbial Technology, IMTech, in Chandigarh, a CSIR India biotech institution of great distinction and repute. After MSc Honours in Microbiology from Punjab University, Chandigarh, he obtained his PhD from the prestigious Indian Institute of Science, Bangalore, in biochemistry. He then pursued postdoctoral research in various prestigious institutions in the USA, including the University of California and the Albert Einstein College of Medicine, as well as the Rockefeller University, New York, where he was also an adjunct faculty. The technology of India's first clot buster drug came from Dr. Sahani's group and his collaborators, followed by its recombinant version, which are now marketed in India as several leading brand names, and are landmark examples of affordable lifesaver medicines in a developing country. He was also the main inventor of India's first biotherapeutic drug molecule, now undergoing phase 2 human clinical trials, which is called clot-specific streptokinase that has been patented worldwide and then out-licensed to a US pharma company. Dr. Sahani and his team then developed fourth generation anti-thrombotic clot blasters that have been out-licensed as very promising leads for ischemic stroke, which remains a major health challenge worldwide. For his contributions, Dr. Sahani has received several prestigious awards such as the Vigyan Ratan Award, the Vasvik Industrial Award, Ranbaxy Award in Pharmaceutical Sciences, the O.P. Basine Award, National DBT Award, CSIR Technology Shield, CSIR Technology Development and Business Award, and various other recognitions. Dr. Girish Sahani is a fellow of all three major national science academies, including INSA. His main devotion has been to help successfully convert laboratory science into usable technologies, and his passion is to inspire young minds to enjoy doing science and take up the challenge of delivering solutions to societal problems. We wish to hear from you, sir, your exemplary journey of achievements, which I'm sure will be a source of motivation for our students. So we request you. Yes.
Thank you. Namaskar to everybody. Good morning. I thank the organizers of this seminar today and the privilege to be a part of this, especially at such a distinguished center of learning, this institute which goes back scores of years and has nurtured so many outstanding leaders, engineers, business visionaries. It is also a great blessing on all of us, and I think I'm very privileged to be here. Not just because I am a Punjab University guy of old, also because today is the birth anniversary of Professor Bhatnagar, who was one of the very few leaders at the beginning of our journey as an independent nation who could nurture science and technology in this country. In the Council of Scientific Industrial Research, still maintains its leadership, its power to contribute to industry and to science. One great example of a young person who has done that is one of the speakers today who was decorated, awarded with a Bhatnagar Award, Dr. Ashwini Kumar, who will speak about his work in tuberculosis today. Bhatnagar Award became synonymous with the best of science and technology in this country. It is also extremely fortunate that we have a person like Professor Grover today who has enlightened us about the heritage of Punjab University. It is a passion with him. He has brought to fore the achievements of scientists, not only all over the country, but particularly in the north of Delhi, particularly. And he is, I dare say, the chief spokesman in this movement. He maintains a stoic leadership of thought and ideas and deep research in this area, and continues to illuminate us and make us proud of our heritage in North India, in Punjab also particularly. I do not want to dwell upon whatever little I did in my career. I got a very generous introduction just now, and I'm very thankful for it. But I want to tell you a couple of things as a person who has done some science, who, just as so many students sitting here in their early 20s, also sat in one of the rooms here in this university and was fortunate to be taught by outstanding teachers. That tradition continues. That tradition also has been bolstered by outstanding research in this university, world recognized. And by all ratings, this university continues to be among the top 5 universities of this country, and I wish it all the success in the future. When you start out as a researcher in your life, there are two temptations. One temptation, undeniable, is to begin a career which is a personal yearning. You want to rise in the world, you want to do well professionally, and as a researcher, you have to manage many fronts. Your fundamental understanding of science and the liberal arts as well, because it makes you a better person, but also manage to run a laboratory, ask fundamental questions, mentor students, inspire them so that they become leaders one day, outstanding performers of the art and science of inquiry. This you have to manage. And sometimes it becomes a little difficult to keep a balance between these two. Personal progress and depth of inquiry. At the end of the day, the lesson I got from my own career was the career should look after itself. You look after your curiosity, your quest for knowledge. And if you do that faithfully, all powers in this world will come and assist you. In my view, humble view, when we get double personalities, when our heart is in one thing, mind is in another, then there is conflict. We have to master that conflict because it is a tangible, all-present conflict in every mind and every heart. So mentoring becomes important. We have to have mentors who generously help you overcome, but not necessarily spoon feed you, but help you overcome the difficulties and challenges from forces from within yourself, and also provide the environment, the logistics, and the convenience of carrying out science. A lot of science, including chemical engineering, most aspect of it, biology, physics, etc., require laboratories, which require funds, and that is also something that has to be respected. There are people who spend their careers doing that and not necessarily get great awards, etc. So this is essentially a teamwork where we have to respect not only our own ambition, but the feelings and desires of your colleagues. That is where the challenge lies. But today, let me congratulate this department and the hosts of this particular meeting, Dr. Sharma, Dr. Gaurav, and many others, Madam Kea, who is such a force with her husband Dharamveer ji of this intellectual activity in this city. I hope that this oration continues in the future. We get outstanding scientists like the two speakers today and many more, and enlighten us and keep the students inspired. And the students have to have a sense of inquiry. And when you look at how people followed their questions, came up with discoveries, as we will do today, it will inspire us and you, particularly young students, to seek out this career, which is not a bed of roses, mind you, which is not an ATM career. It will not give you huge amounts of money, as many other professions would, but it will give you one thing. It will satisfy your curiosity. The primary urge in every human heart is know thyself, know yourself, know this natural world, this creation. You cannot understand all of it, but in one lifetime, you can understand some of it. And when you understand some of it, you feel fulfilled. Thank you very much.
Thank you so much, sir, for your enthusing words of wisdom. I'm sure the students have learned a lot from your words of wisdom, and they'll imbibe these words in their life. So I want to take this opportunity of having stalwarts of sciences and research in our department, and we would like to request Dr. Sahani to release the book on nanostructures authored by Professor Gaurav Verma of our very own department. Congratulations, Gaurav Varma, sir. I would also request Professor Gaurav Varma's family members to come on the stage for a photograph. And Professor Arun Grover, sir. Congratulations, sir, and it is an honor for our department also that our professor has authored a book of repute. So I would now like to request our chairperson to felicitate Dr. Girish Saniji with a token of appreciation. Thank you, ma'am. Thank you, sir. Now let's proceed, and it's my pleasure that the alumni spanning the pinnacle is always an enduring moment for the faculty, staff, and students of any department. I take privilege of introducing Sri Sanjay Arora ji, DCT alumni of 1987 batch. Sri Sanjay Arora ji has done BTech from DCT and also holds an MBA degree in marketing. His first job was with Punjab National Fertilizer Chemical, Naya Nangal, Punjab, as a production engineer. After serving with PNFC for 2.5 years, he joined Yale in 1991 and is continuing with the same company. In Yale, he mainly handled plant operations and commissioning of gas processing plants, petrochemical plant operation, and gas cracker project. Currently, for the past decade, he has been overseeing the gas marketing function at Yale in the Chandigarh zonal office. With a wealth of experience, he has accumulated a total of 36 years in the industry. We once again welcome you as the guest of honor for Bhatnagar Day Oration and would request you to share a few words of wisdom.
Very good morning. Dignitaries sitting on dais, organizers of this Shanti Bhatnagar Institute, faculty members and younger generation students. Very good morning. In fact, just a few days back, I got a sudden call from Professor Anupama. Normally, in office, when basically these unknown calls are usually coming, loan ले लीजिए या कुछ credit card ले लीजिए. So suddenly I picked up, and that was a blessing in disguise, in fact. Yes. So thanks, Professor Anupama, for inviting me. I'm really privileged to be part of this event. मेरा बेसिकली इससे काफ़ी अटैचमेंट रहा इस इंस्टीट्यूट से, क्योंकि मैं बेसिकली डे स्कॉलर ही था और आई यूज्ड टू कम फ्रॉम सेक्टर 23 चंडीगढ़ बाय CTU bus. So that time even टू व्हीलर वाज आल्सो a dream. उस टाइम मारुति की बुकिंग भी शायद शुरू नहीं हुई. So बहुत अटैचमेंट रहा चंडीगढ़ से। तो इनफैक्ट जब पास आउट हुए तो ऐसा लगा कि केमिकल इंजीनियरिंग की है तो किसी ने सही बोला था कि इंडस्ट्री में ही रहना पड़ेगा और बेसिकली शिफ्ट ड्यूटी करनी पड़ेगी। एग्जैक्टली वही हुआ। और उसी में ही बेसिकली ये था कि चंडीगढ़ के जितने आसपास रहेंगे वो आई थिंक एक्सपीरियंस भी मिलता रहेगा और जुड़े भी रहेंगे घर से। सो नंगल में ज्वाइन किया। बेसिकली उस टाइम ऑटोमेशन कुछ खास था नहीं, तो बेसिकली मैनुअल पीएलसी कंट्रोल होते थे, स्लरी हैंडलिंग सिस्टम होते थे। उसमें फिर आई कैन रिकलेक्ट कि वो बेसिकली स्लरी हैंडलिंग में क्या होता है कि चोकिंग होती थी लाइंस में। सो वी यूज टू मैनुअली पोक द लाइन एंड क्लियर द चोकिंग। सो दैट वाज द पीरियड। देन at that time when I left PNFC in the year 1990. So basically, uh, then I got the opportunity in one of the leading this gas transportation and trading company, GATE India Limited, which is now Maharatna company. So I joined same जो वहाँ पे गैस ऑपरेशन और ये जो कमीशनिंग पीरियड करना पड़ा वहाँ पे भी। So that was the time when इंडस्ट्री वाज ग्रोइंग। हमारे पास उन दिनों लिमिटेड ही ऑप्शन थे, रिफाइनरी कम थी, लेकिन गेल ने काफ़ी अच्छा ग्रो किया। दी गेल नाउ इट इज़ ए लीडिंग बेसिकली ट्रांसपोर्टेशन कंपनी। हमारा जो क्रॉस कंट्री पाइपलाइन का नेटवर्क आज की डेट में 15,000 किलोमीटर है, जो कि एक महारत्न कंपनी में बेसिकली वी आर स्विचिंग टू CGD sector, city gas distribution also, and renewables also. Recently, we have signed the agreement with Punjab Energy Development Corporation for setting up of a compressed biogas plant. So GAIL has been this a very good journey. I have worked in operation department, then projects department. Now from last 10 years, I am with zonal marketing. So I have seen over the years GAIL has grown quite good. Now the 36 years I am there in the field industry. I have worked in corporate sector. So my advice is that basically for younger generation that as आप जितना भी स्ट्रेस फ्री में रहेंगे उतना ही बेसिकली आपके कैरियर में ग्रोथ दिखाएगा, जो कि बेसिकली इतना इजी नहीं है। लेकिन मेरा जो एक्सपीरियंस जो अभी तक मैंने देखा अपनी लाइफ में या अपने प्रोफेशनल कैरियर में, सो अवॉइड स्ट्रेस एज फार एज पॉसिबल। दिस इज द फर्स्ट एडवाइस। देन बेसिकली आप एक टीम में अगर काम करेंगे तो टीम से अगर डिसेस हो गए कभी भी, सो उसमें आपको टेम्परेरी गेन मिल सकता है, लेकिन परमानेंट गेन के लिए आपको अगर सक्सेस देखना है तो एक टीम में काम करिए। और हर हर कोई यही चाहता है कि बेसिकली पोजीशन चाहिए हमें। तो पोजीशन लेने के लिए आप बेसिकली पा सकते हैं पोजीशन, लेकिन स्ट्रेस फ्री रहेंगे और बेसिकली आप अगर ग्राउंड से जुड़े रहेंगे सो इट विल लीड टू ए सक्सेस। थैंक यू वेरी मच।
Thank you, sir. I express my sincere gratitude to you, sir, for sharing such inspiring and impactful words with us. I'm sure everyone agrees that today mental health, dealing with mental health, stress management, and team spirit is the mantra for success for the young generation. So I take the opportunity again today is indeed a joyous occasion. We are going to release our first departmental newsletter, which is possible because of the efforts of the chairperson, faculty, and students of Dr. SSBUICT. This newsletter covers all the academic, technical, cultural, and sports events organized by the department. For the release, I request Sri Sanjay Arora ji to do the honors. He is an alumnus of DCET. And I request all the faculty members of the department to join on the stage and release the book. And the students who have worked for this newsletter, please come on the stage. Join us on the stage and we'll take a group photograph.
I request Kia ma'am also please to come on stage.
Adding to the achievements, we take great pride in unveiling the second edition of Jeevanam, a magazine dedicated to food technology. Jeevanam is a Sanskrit word meaning pertaining to life. The first edition of Jeevanam was released in November 2022. It is noteworthy to mention that the articles presented in this magazine are from the students of all the streams of Dr. SSB UICT. Jeevanam attempts to explore the various aspects related to food, the advancements that are transforming the way food is processed, packaged, and delivered, ensuring optimal food safety and high quality standards. I once again request Sri Sanjay Arora ji to unveil Jeevanam along with all the faculty members of the department, especially the food technology students and faculty, and Professor Arun Grover ji. Please come on the stage, and our alumnus of department, please come again on the stage for unveiling of Jeevanam. I request all the faculty members to join again on the stage. And we need a group photograph in an organized way now. Yes, all ladies in front.
भरोसे पीछे हो गया
Thank you.
I would now request Chairperson Ma'am to felicitate Sri Sanjay Arora ji. Chairperson Ma'am, I request you to felicitate Sri Sanjay Arora ji. Thank you, ma'am. Thank you, sir, for your esteemed presence today. Without any further ado, let us concentrate on the focal point of today's event, which is the Bhatnagar Day Oration talk by Dr. Ashwani Kumar. I take privilege in introducing Dr. Ashwani Kumar. Dr. Ashwani Kumar is an Indian microbiologist and the senior principal scientist at the Institute of Microbial Technology in Tech. He is known for his studies on Mycobacterium tuberculosis, the causative agent of tuberculosis. His laboratory focuses on understanding the reasons for drug tolerance observed in humans. His laboratory hypothesizes that tuberculosis is a biofilm infection, so its treatment needs the administration of multiple drugs for at least 6 months. He has been bestowed with numerous awards and recognitions. To mention a few, the Department of Science and Technology has awarded him with the Swarn Jayanti Fellowship for 2016 and 2017. The Department of Biotechnology has awarded him with the National Bioscience Prize 2017-2018. He was selected for DBT Wellcome Trust India Alliance Senior Fellowship from 2021 to 2026. He was elected as a Fellow of the National Academy of Sciences India in 2022. For his contributions in tuberculosis pathogenesis, he was awarded with Shanti Swarup Bhatnagar Prize for Science and Technology 2022. He is considered one of India's leading scientists in the field of tuberculosis, and his lab is doing some of the best research in India. I invite Sir for today's talk on mycobacterial biofilms. A new understanding paves the way towards shorter treatment of tuberculosis.
Namaskar, Professor Grover, Dr. Sahani, Professor Sharma, Professor Gaurav, Professor Mohsin ji, Arora ji, colleagues from Punjab University, and students. It's really a great honor and privilege to talk about some of our work to commemorate Professor Dr. Shanti Swarup Bhatnagar. And today I'm going to be talking about tuberculosis, which is the area where we work. Just give me a minute so that I can get the laser pointer. Yeah, so we're going to talk about tuberculosis. And Dr. Sahani recruited me at IMTech in 2010. So it's really a big thing for me to present some of our work in his presence. And in this journey, I have been accompanied by these students, Abhishek, Oshali, Sapna, Parminder, and Shweta. And they have made this work possible. So I would like to start by acknowledging them. And I will primarily talk about tuberculosis, give you a glimpse of what we do in the lab. and how we go about it. So how many of you know about tuberculosis? No, not many. I'm pretty sure you would have heard about it. Amitabh Bachchan ji does advertise about it, about the treatment. We'll go about a few things. If you don't know, probably by the end of the lecture, you will learn a bit about it. This is my lab. We work on a lot of things. Only one part of that is biofilms. This one. And I'm going to talk about that. So let us talk about tuberculosis. Tuberculosis is a disease. It's a big disease actually, and we characterize this disease by, let us say someone is having cough, fever, have weight loss, at least for 3 weeks. Then they go to the doctor. Doctor give them a cough syrup and an antibiotic. It does not work. They go again to the doctor. Then doctor asks them to get a chest X-ray done, and with the chest X-ray we figured out, yes, there is a scar in the lung which indicates there is tuberculosis. So by the time somebody feels symptom, it already takes around 1 month's time to diagnose that there is possibility of tuberculosis. Many a times chest X-ray is also not positive. Then you have to go for culturing. So culturing is difficult. Culturing it takes at least 1 month for the colonies to appear. So it will take 2 months for diagnosing TB. If there is too much of load in the sputum of the bacteria, this bacterium is called as Mycobacterium tuberculosis. It is the main agent which causes this tuberculosis. And if there is around 10,000 bacilli per ml of sputum, then you can detect it on a smear, which we call as acid fast staining. So it takes quite a lot of time to really diagnose the disease after showing the symptoms. And let us say somebody is exposed to this individual, that person will not become immediately diseased with TB. That person will take around 1 to 2 years to develop tuberculosis, suggesting that tuberculosis is a chronic infection. Many times, a child who got TB from his father will show up it in 40s. So TB will be latent for this long in that individual. So we call it, once you are infected with TB, you are infected for life. Most of the people carry it, and when they become older, they can have the disease with the same bug. Many times there is recurrence of infection, but that is we are not going to talk about that today. So this form of TB, which is very prevalent. We call it pulmonary tuberculosis. There are other forms of tuberculosis which are difficult to diagnose, even more difficult than what I described right now. And these are called as extrapulmonary tuberculosis infections, and they can be in different parts of the body. Somebody can have it in meninges in the brain. We call it TB meningitis. Somebody may have it in bone. Somebody may have it in skin. Somebody may have it in colon. Somebody may have it in kidney. So there are whole plethora of mycobacterial infection which can manifest itself in the form of extrapulmonary tuberculosis disease. The only saving grace with this is that this is not communicable. The pulmonary tuberculosis is communicable, while this is not communicable. So that is a good thing. Let us look at how does the situation look in 2020. In 2020, we lost 1.5 million people, and 1/4 of that was in our country. There were around 10 million infections. In 2022, the situation is same. In 2020, it was the single largest infectious killer. In 2022, it became second only to COVID. And trust me, COVID came and it went. Tuberculosis has been there for centuries now. It is the leading cause of human death in the human history. It's a very old disease, it's a very old enemy. We can find the evidence of tuberculosis in Egyptian mummies, in Hungarian mummies, in Colombian mummies. And one of the distinguishing feature in these mummies, I guess you will be figuring out, is something like a hump in the back. This hump is because of tuberculosis infection in the spine. And we can recover the tuberculosis DNA, mycobacterial DNA, from the bones. We can scrape the bones and we can get the DNA, and we can even say, at times we also find RNA, which is the long stable RNAs which are there, which still can be mineralized and can be recovered. So this is a very old disease. In fact, evidence suggests that this bug has been evolving with humans, and its evolution is marvelous. That is a topic for another day's talk. It evolves by losing genes rather than by gaining genes. In fact, its cousin brother In soil, it has only two-thirds of the genome. And its cousin, leprosy, it has half the genome of MTB, and leprosy was also another big disease. I am pretty sure you all know about Manthara in the Ramayana, and she had a hump, and that hump is suggestive of either cured TB or the spine TB, which we also call as Pott's disease. So this has been there. We know it very well since in all the history. It is described in Vedas as Raj Yakshma. Raj means Raja, Yakshma means disease. So it has been known as the king of the diseases. Even Bible describe it as Sishifit, which means a disease which consumes humans. And in 18th century and 17th century, this was the leading cause of death in Europe. And one-fourth of all the deaths above 12 years of age in humans between 1800 to 1850 were dedicated to TB, and it was also known as white plague because of this much of the disease. What is the situation today? So this is how it looks like today. I'm sorry about this slide has got a little kind of weird, but this is the situation today. One-fourth of the world's population is latently infected with TB. I told you, once infected, always infected. So one-fourth of the world's population is infected with TB. It is more so in a country like ours and less in Western countries like Europe and US. In this room, I'm pretty sure if I do some analysis, we will figure out that around 60 to 70 people, 60 to 70% people will have TB infection. They all are healthy. There is no disease, but they're infected. I am also infected. So are many more. Unfortunately for us, India happens to be the epicenter of this disease. Of total deaths and total cases in the world, we lose around one-fifth of, uh, uh, we have one-fifth of the disease burden in our country. And you can see with this big blob in the country. So to tackle every disease, you need three things. The first is diagnosis, the second is a vaccine, and the third is the treatment. Vaccine is the most effective method, but let us see how does it get diagnosed. I have told you a bit. We use methods which are 100-year-old to diagnose TB. We go with the classical acid-fast staining and looking at a microscope. And if you go to tertiary care centers, there are bigger machines, but at the primary care centers, you will see microscopes not even having LED as the light source, but the sunlight as the light source. And the sensitivity of these assays are very little. We only diagnose between 35 to 65% of the cases that are there with the help of acid fast staining. Culturing is more sensitive, it is gold standard, but it takes much longer. And X-rays we have been using since 1930s, and they have been at the forefront of diagnosing TB. The second thing is vaccine. Let us look at the vaccine. Vaccine was developed by Calmette and Guerin, and is called as Bacilli Calmette Guerin. It was given in 1921, and it is the vaccine that has been administered as it is the vaccine which has been administered most in the history of humankind. Despite that, this is not really very good vaccine. It only protects children to some extent from TB. Children's, because they don't have immune system, they can have biliary TB, which can be very deadly, but it does not really protect individuals beyond age of 10. I'm pretty sure if you look at your left arm, you might see a scar there. That scar is because of BCG. BCG is given in such a way that it leaves a scar because of the hyperimmunity, hypersensitivity reaction, and that is there, which means you all are vaccinated. Despite that, we get infection. So the immunity that is generated by BCG does not really go for long. Let us also look at the treatment. How does the treatment happen? This is the prescription of an individual who was having fever, and this individual has to be prescribed with only one antibiotic for 3 to 5 days. That is easy. And this individual was infected with the TB, and it has to be prescribed for 4 antibiotics for 6 months, and this is called as short course therapy. Directly observed short course therapy by WHO, and the short course is 6 months. Earlier, it used to be 9 months to a year, depending on which physician is prescribing which medicines. So there is a problem. The problem is we have to give these many drugs for very long period of time. So there are compliance issues. If I tell somebody to take something, even water every morning, they will forget sometime, and that's a problem that leads to drug resistance TB. So whenever the resistance is against one drug, rifampicin and isoniazid, this is given in a combination. We call this at MDR-TB. When there is drug resistance to fluoroquinones and second line drugs like linezolid or bedaquiline, we call it extensively drug resistant TB. And there is a new variant which is called as totally drug resistant TB, for which we have no antibiotics to cure whatsoever. And this treatment is pathetic by every measure. Why? This is a simple pill, and a patient with MDR-TB has to take 14,600 pills. If I stack them one by one, the length of that will be 3 times that of Qutub Minar. I'm pretty sure everybody knows Qutub Minar. So that many medicines these individuals have to consume, and this is the success rate. The drug sensitive TB, if we treat them with all the available drugs, 83 to 90% success is there. They will complete the treatment within 6 months, and 83% of them will be healthy. We lose around 10 to 17% individual. So we always say pura ilaj pakka ilaj. It's not true, but we have to say it. The second thing is MDR-TB. The treatment goes for 2 years, and only 52% individuals are able to complete that 2 years. We are not saying cured. They complete those 2 years. The rest of the 48% we lose. And if we do XDR-TB, the treatment is longer. Only 28% go through that treatment of 2 years. Rest are lost. So we need drugs. We need to figure out what is going on with TB if you really want to conquer this enemy. And trust me, the drugs that are there, they were discovered although in 70s, 60s and 70s, but they are good. If I take a culture, this is the culture tube, and if I treat the bacteria in there with combination of these drugs, let us say with rifampicin or isoniazid, I can kill mycobacteria very fast. Within a week, most of the bacilli will be dead. But if I put the same bacilli in the animal, let us say guinea pig or mice, I give these drugs and they will not have that much of efficacy. You can see that in mice, they are more working and most of the testing is done in mice. In guinea pigs, they work even less. In rabbits also, they work even less. So there is some dichotomy here. The drugs which are very good in the culture, they are not able to kill in the animal. There is some physiological thing that we don't understand. There is some problem there. And we have been working on this pathogen scientifically for last 150 years, and we still do not understand the biology enough to kind of shorten this treatment. So there are two hypotheses around this. The first hypothesis is that there are persisters. So persisters are nothing. You give drug treatment, some of the cells die. There are others which will not respond to drug. Those are called as persisters. So and then there is a biofilm. The persister is a very old hypothesis. It was given in 1940s. Now the connotation of persister has changed. It is only talked about with respect to drug. Earlier, when it was discovered in tuberculosis, the connotation was with respect to the host. So there were bacterial cells which were going to survive in the host for a very long period of time. They were called persister. Now we call persister those bacterial cells which can survive in the presence of drug. So the biofilm hypothesis is a new one, and Graham Hatfield's lab and our lab has been kind of going for this hypothesis. Initially, there were hurdles. People were not accepting this hypothesis. Now it is more accepted. So in this hypothesis, what happens is that initially few bacterial cells adhere to a surface, and then they start talking to each other and talking to the other bacterial cells. They invite others to come at this site, and then others come, and then this bacteria is not a single individual. This is a multicellular kind of organism where there is a differentiation of work amongst these bacterial cells. There is cooperation amongst them, and they all make together this big castle kind of thing, which we call as biofilm. So in this castle, the macrophages, which are one of the main cells which attack tubercle bacilli, they are not able to get in there. So they are protected. Bacterial cells are protected. Even drugs are not able to get in there, and they are protected. But this still is a hypothesis. Fortunately, the recent NIH data suggests that 80% of chronic infections, they are biofilm infections, and they are at different diverse sites in the human body. And these are really difficult to treat because, as I told you, the drugs do not reach them. The immune system, the immune cells, they do not reach them. So they are really difficult to treat. And biofilm is basically the natural way of a bacterial cells. And for last 100 years, we have been kind of studying the bacterial cells in their unnatural state, where we grow them, give them food and everything, and then kill them with antibiotics. So the planktonic cells which grow in the tube looks like this. They are easy to treat, they are easy to kill, they are easy to get by the immune system also. Whereas when they come together, make a community, they are difficult to kill because you can see that macrophages cannot get to them, and there are mechanisms through which they can they do not allow the antibiotics to reach them, and they keep a reservoir of food and other things with them. In fact, lot of drug resistance genes, they are passed from one organism to another in these structures. So that is important. Let us see what can these biofilms do. So these biofilms help in aggregation of bacterial cells, provide adhesion, provide phenotypic heterogeneity, which will play a role in the drug tolerance. And the major hallmark of these biofilms is their EPS, extra polymeric substance, the substance in which these bacterial cells are there. And this substance is really the key to understand what the biofilm is. So if we understand the EPS of these biofilms, we can perhaps figure out ways to disrupt these biofilms, and then the drugs and immune system will reach there faster. But in MTB, this has been neglected as a hypothesis. We know that biofilms are there for 1970s, in fact, earlier than that. But in 70s, we started to see biofilms during infections. But this TB field always rejected this hypothesis for one major reason, that Mycobacterium is an intracellular pathogen. And for making biofilm, you need large number of bacterial cells to come together and work together. And this is how mycobacteria looks like. There are only few cells, and these 4 or 5 cells maybe cannot make a biofilm. So for this reason, this hypothesis has been discarded altogether throughout the history. Then there are few labs like ours who still believed in this hypothesis, and this is a biofilm which is called as pellicle biofilm. So this is a growth of mycobacteria at the liquid air interface, and if I take these cells and I treat this culture with antibiotics, antibiotics will not be able to kill it. And my student Abhishek, he developed another model where he treated mycobacterial cells with the redox reagent called as GTT. I'm pretty sure many chemists and biologists use that in their labs. And what he found that the biofilms can be made within 29 hours upon exposure to DTT. And this is important because this makes now this a tractable model where we can study the bacteria, how it changes, what kind of polysaccharides are found very quickly, compared to the traditional model, which takes around 5 weeks to make these biofilms. Then initially there was enigma, how does this reductive stress leads to the formation of these biofilms. Later on, my students Parminder and Shweta, they came together and they demonstrated through series of experiment that is published now, that upon reductive stress, two things happen. First is that the BSA in the medium that becomes reduced and start to aggregate, and with which the bacterial cells interact and make micronuclei through around which the whole biofilms are developed. And once the biofilms are developed, then we have drug resistance. So we also tested whether the biofilm model that we have, whether that has drug tolerance or not. And here is to show the data with isoniazid, INH. These are the frontline drugs. All these drugs used at 1x or 10x were not able to kill the bacteria residing in this biofilm. Not even 1% of the bacilli were killed. But trust me, the data here is only for 10x. We have used up to 1000x for 7 days, and the drugs are not really able to kill the bacilli residing in this biofilm. So bacteria is in different kind of physiological state where it does not respond to the drugs. Then Abhishek did several experiments, and one day he showed me this data. The data was very clear that this is the biofilms, and we treated biofilms with different enzymes, many different enzymes. And what was surprising to us was that whenever we treated biofilms with cellulase, the biofilm got disrupted. Cellulase was difficult initially I did not even trusted the data, but then he kept on doing the experiment, and every time it was reproducible. So that kind of convinced us that maybe there is some cellulose, because cellulase is an enzyme which degrades cellulose. Cellulose is the plant material that we use. These clothes are cellulose, the wood is cellulose, and it's one of the most stable and biological material which has a lot of tensile strength. The other thing was that there are some proteins that are perhaps playing a role in the maintaining the structural integrity of these biofilms, because upon treatment with proteases also we could disrupt the biofilms. The identity of these proteins, these structural proteins or the anchoring proteins, is still not known, and cellulase Abhishek was able to demonstrate. He also showed that when you disrupt these biofilm using cellulase, then the drug sensitivity is reinstated in these bacterial cells. Let me tell you a bit more about cellulose. So cellulose is the plant cell wall material. It's there in the plant cell wall, and it's synthesized through this enzyme complex, which is consisting of BS, bacterial cellulose synthase A, bacterial cellulose synthase B. There are other enzymes which play a role in this, and then together they make these kind of microfibril-like structure. And the cellulose that comes from plant is crystalline, and the cellulase that bacterial cells may make for biofilm is amorphous because it has to retain a lot of nutrients for helping the bacterial cells. So, because it was kind of enigmatic, we did a lot of experiments to be convinced ourselves and to convince others that indeed cellulose is there. We purified the cellulose from these biofilms, characterize it biochemically, and this is just to show you that the purified cellulose look like this, and we can do FTIR analysis, PXRD analysis, and many other analysis to prove that it is indeed cellulose. And we could also stain it inside the biofilm using a dye called as calcofluor white. So you can see that between the microcolonies there are cellulose which are kind of holding these biofilms together. Then Koshali joined the group, and she showed that not only in Mycobacterium tuberculosis, in whatever species of Mycobacterium we have taken, all of them make cellulose, utilize cellulose for making these biofilms, and we use different models of biofilms. So in all of them, cellulose was a universal thing. The next thing was whether this can be physiologically relevant. So we studied the literature, and in literature you find that the notion that Mycobacterium tuberculosis is an intracellular pathogen is not true. There are a lot of bacterial cells which are there in clumps or bunches which have potential to make biofilms. Literature was filled with them with different species of Mycobacterium. Some papers were also indicating that Mycobacterium tuberculosis can do so also. So, Posha, along with and Shweta, they worked on this that we can make the granuloma, which is the hallmark of TB, inside the laboratory in a test tube. And we made that, and there we found cellulose was there. Then we did mice experiments. I'm not showing those experiments here. We did non-human primate experiments, and all of them we found the biofilms where the MTB were encased in a cellulose matrix, and we concluded that they are biofilms. And then the data I'm showing here you is from humans. We got the human samples from PJMR Chandigarh with the help of Dr. Radotra. He's retired now, but he helped us a lot in this project. And this is how the granuloma looks like in the human lung. And this is how the bacilli, few of the bacilli that are detected looks like. So we checked for the presence of Mycobacterium biofilm, and we utilized two things. First was the calcofluoride, which I have shown you earlier. We developed another probe which we call as CBD cherry. So this can specifically stain the cellulose. So we found that in a lot of lung lesions, we find that bacteria is surrounded by cellulose. We concluded that these are biofilms. If we treat the lung block with cellulase, we can see the release of glucose, and we can perform confocal Raman microscopy to show that around mycobacterial the characteristic bond of beta 1 fold linkage, which is there in the cellulose, is there. So all these data convince us that indeed there are biofilms in the lung of humans and other models that we use. Then Sapna and Koshali, they grouped together and they constructed this strain by overexpressing a cellulase in Mycobacterium. And this is what is interesting in this strain. This is the wild type, and these are the strains that are overexpressing cellulase, two different cellulases. These strains grow as normal as the wild type, but they are only deficient in one property, that is to capability to make biofilm. So they are not able to make biofilm, and this is shown here. And their colony morphology, which is another kind of biofilm, looks very different. And this is how their lungs look like. So this is the infected lung with TB, and this is the infected lung with the bacterial strain, which is overexpressing active cellulase. This is slightly less active cellulase, so there are some lung lesions here. But this lung looked very healthy, suggesting that The capability to make these biofilm is central for mycobacteria to establish an infection in the mice. And when we did CFU, we found that if upon giving 100 CFUs, the bacilli would only reach to 1000 CFU, which overexpress cellulase. So, to establish an infection altogether, mycobacteria needs to make this biofilm. These data suggested that TB, indeed, we have been reading it wrong. TB is in fact a biofilm infection, and because of that, it is chronic in nature, and because of that, we need long treatment for this drug, for this disease. Then Poshali and Sapna did a crazy experiment. I call it crazy experiment because initially I was not even convinced that this can really give some data. So they made this kind of a chamber in which they made mice to nebulize cellulase in addition to the drugs that are given to the mice. And this was the experiment. So a group of mice inhaled cellulase in addition to the drugs, and a group did inhaled heat inactivated cellulase along with the drug. And to our surprise, this was we have done it for 4 weeks. And to our surprise, the group which has received nebulized cellulase has better killing of mycobacteria. This is very significant because if I just give isoniazid or rifampicin alone, I see around half a log difference. When we give them combined, then we see around a log difference. And getting a half of log difference just by adding something in the nebulization is very, very good. So, what is the significance of this study? There are two significances. First is that mycobacterium is a biofilm infection. So far, we have been treating it wrong. The second is we may develop things like cellulase, which can chew up the polysaccharide material and make bacteria more amenable to treatment with drugs which are already there. Thankfully, our science was covered in the journal Science. They wrote a commentary on it, and now cellulase is regarded as a virulence factor in the field. Soon after this, a paper got published uh, where they talk about the presence of these biofilm like cords in the lungs. So our data gets validated. We have said that there is biofilms in the lung, and they show they call it biofilm like. They are apprehensive that to want to call it to biofilm, and they have seen these cords, beautiful cords in the lungs of mice. And then we looked at the cords, and we found that these cords also have cellulase. And if we use a strain which is overproduced cellulase, they are not even able to produce these cords. So all these data together suggest that same thing which I told earlier, that TB is a biofilm infection, and we can create new methods to disrupt these biofilm to treat tuberculosis better. And that's the end of it. In the acknowledgement, my lab is very well funded by different agencies, and I'm thankful to Dr. Radotra, Dr. Deepak Vashal for their help in the research. And this is my lab. Some of the people are sitting here. Thank you. I'll be happy to take questions.
Any questions? Sir, I have a question. I don't know, it just came to my mind. Any person who has a hump doesn't mean it has either libido or.
No, it does not necessarily mean, but in endemic countries like ours, if somebody has a hump, 80% chances that individual either has a tuberculosis going on or there is a cured tuberculosis. There is a 80% chance. So it is like in our country, if you see mediastinal nodes swollen up, we are going to call it tuberculosis. In West, if you see, we are going to call it sarcoidosis. But in here, if somebody has a hump or something, the most prevalent infection of the spine is tuberculosis.
So I've seen my parents as they age, especially my mother, she's been growing a hump in the back, but she's never complained of any issues. So does that mean we need to go and get it checked? So you can see that, and it is so very apparent.
So the hump keeps growing because of the inflammatory reaction. If that hump is growing, then definitely we need to see
Yeah,
that.
I can see it growth in the last 5 to 7 years. I mean, it's not a quick.
Yeah, mycobacteria is known for that. Mycobacteria is very slow. So even causing the disease or symptoms to come up, it takes around 2 years.
I really want to, congratulate you on an amazing science that you and your group have done. I have a few questions. The first one is, so you mean the biofilms are growing outside the macrophages? The mycobacterium is not intracellular at all?
No, so there are two possibilities. The first is that there are extracellular bacteria which we have ignored completely so far. We believe that is going on. Besides that, now new data in the Cell paper that appeared this year, last year, end of this year beginning, they show that there are these cord-like structure even inside the cells. So for Salmonella, we know that in the macrophages it can make biofilm. There are only 5-6 of them, but they start going in this kind of EPS, which we call biofilm. And for TB also, we are of opinion that even in the macrophages there are biofilms.
So you mean 5 or 6 of them make a biofilm, which gives them the resistance?
So the new data that comes in, earlier we used to believe that around 15 bacilli per macrophage will kill the macrophage and come out. But now the new data suggests that mycobacteria can grow in larger numbers than that. And the data from Vivek Thakkar's lab shows that we can have many more bacilli and they are held together with EPS. They are not really inside the phagosome, the classical niche. They are really outside that, and that is how they have access to cytoplasm even.
So maybe the coral sensing is also at a lesser number.
Yeah.
Okay. So anywhere you came across in literature or any studies on the lipid substrate, like you talked, there is a cellulose substrate there, and we always know that mycobacterium are lipid-loving bacteria. So any data on any lipidic substrate?
Yeah. So before us, there was 2000 in 2005. There was Cell paper where the group of Graham Hartful suggested that there are short chain mycolic acid which are not part of the cell wall. They come out and they make a sea of lipid in which these hydrophobic cells float, and that is what was called biofilm. We have not found evidence of secreted short chain mycolic acid in our studies. We treat the biofilms with lipases and the biofilms are intact. So we do not believe that. Initially, that's what everybody believed, but now more and more people believe that polysaccharides are the major component of the EPS, the matrix, and not the lipids.
All right. So I'm basically a formulation scientist, and I did a lot of work initially on all the conventional antitubercular drugs where I tried to encapsulate them into solid lipid nanoparticles. And one of the hypotheses, it's been several years now when I stopped working on them. So one of the hypotheses at that time was I was working with solid lipid nanoparticles, and we did show a lot of improved pharmacokinetic studies at that time because all of them are hydrophilic. And then we did some studies with mycobacterium stigmatis because we didn't have the DSL 3 or 4 labs available with us. And we could show that there is a lot of improved sensitivity of these to evenly work with some resistant strains. And the hypothesis we proposed was that since they are lipid loving, so they pick up those SLNs as a food for them and then end up being killed by the drugs which enter them. We have seen a lot of antibiofilm and biofilm dispersal with some of our nano particles in fungal biofilms and some bacterial biofilms. So I was wondering that maybe there could be a role here also.
Yeah, so that is important. So lot of pharmacokinetic and pharmacodynamic studies on drugs are coming out. Fortunately, the drugs that we have, isoniazid, pyrazinamide, ethambutol, now bedaquiline. So there is one unique phenomena with them. Although very little reaches in the granuloma, but they accumulate inside the mycobacterium. So once they go in the mycobacterium, they do not really come out. There is a lot of data coming out now. So because of this affinity of the drug to go inside the bacterium and staying there, accumulating there, that leads to concentrations which can kill mycobacterium. So already very little drug is reaching in the granuloma where the bug is. And if we can increase that concentration, the drug that will accumulate in the bacterium will be far more, and then we can kill it better, for sure.
Thank you.
from the your speech, your interest, your deep study of the whole. What I would like to ask is, are you planning to do any success in finding some method to overcome this disease?
So I have shown some data with the mice. We need to do more work. This just came out. Uh, that if we can disrupt these biofilms, we can shorten the treatment. We have to look at the relapse rate, which we have not done in this study. So the promise that this study brings out is that if we can figure out ways of disrupting the biofilm using cellulase or small molecule agents which can disrupt these biofilms, then perhaps we can imagine a situation where TB will be treated in a month's time rather than 6 months. But it is, I would say, around 10 or 15 years away. It's not right there right now because the trials of TB are very long. So even if a vaccine today enters the clinical trial, it will also take that much of time. The TB trials are different than other diseases, but we see there is no interest. People are now putting biofilm model for screening the drugs, which was not there earlier. So we believe that in another few years, this science will really get translated. So, unfortunately, the pharma industry is not really keen on making new drugs for TB because if you treat TB in few days, it's not much of business. But if you make a drug for diabetes, for which the individual has to take drug for 40 years, that is more of business. That is the unfortunate thing. But fortunate, there are many good things also. NIH, FNDR, there are many organizations which have come together, Bill and Melinda Gates, and they have supported efforts to have new drugs. And bedaquiline was approved by FDA in 2014, and that was after around 50 years that a new drug was approved for treating TB. And now there are two more drugs which are in clinical trials and which are given to patients with MDR TB because they don't have any other resort, Pretominid and Delaminid. So that is the good news. Uh, we also work on drug development. So our dream is to reduce from 4 pills to 1 pill. 1 pill, like we treat other infections, 1 pill which can kill it. And our target is bioenergetics of mycobacteria. So how the bacteria gets energy. We are trying to work on that. Let us see in another few years if we can get something.
How does a normal human body, for most people who do resist this, how is it that we are able to resist this?
So the good thing is that of 100 people infected, only 5 will become diseased in their life, 5 to 10. Rest 90%, we don't know the mechanisms. We don't know.
Maybe that would be
The,
for you
yeah.
to get the answer.
Yeah, but that is some that expertise I don't have. The immunologists have that expertise.
I'm not a biologist. You said that non-pulmonary types of tuberculosis are not communicable. So how does one get it in the first place?
Okay, so so there are this I'm talking of 1950s when these experiments were done to figure out how does and where does the bacteria go. So these experiments were done like this: that you inject MTB in the tail vein and look which tissues will get mycobacterium. So the first tissue that got mycobacterium was with liver and then in lungs. But besides that, now there is data that in adipose tissue, in even stem cells, mycobacteria go and reside. So what happens is these individuals have inhaled mycobacteria, and from lungs, mycobacteria has moved to other sites in the lung because of the immune system was not able to cause a disease, but it has, let us say, moved to kidney. And in kidney, it is able to make a niche and cause the infection there through the blood vessels. But now it cannot come back to the lung and spread because the disease is localized in the kidney, let us say, or in the spine. The classical example of this is something called as TB pleurisy. So pleurisy is the cavity in which our lungs are there. So it's a kind of membrane around the lungs to protect them, and there is a liquid filled in that. So many a times bacteria is not able to cause disease in the lung, but in that cavity. So even that bacilli is not able to move into the lung and spread to others. So these are non-communicable form of diseases, and the only problem is they're difficult to diagnose because the samples are not readily available, and treatments are often longer. I guess there are no more questions. Thank you. Oh, there is one.
Name is Unity, right? He will still be with us in 1 o'clock.
All right, thank you.
This calls for a huge round of applause for such an insightful presentation by Sir. I am confident that it has illuminated everyone in attendance. We express our sincere gratitude for your articulate delivery of explaining the causes, diagnosis, treatment of tuberculosis. Thank you, sir. Now I would request Professor Anupama Thakur ma'am and Professor Arun Grover ji to please felicitate Dr. Ashwini Kumar. Thank you, Dr. Ashwini Kumarji, for such an insightful presentation explaining your exemplary work in the field of tuberculosis. And now we request everyone to join us for a cup of tea. And we want all of you to join back again in the auditorium in 10 minutes because we again have a very interesting talk by Professor Hussain Remadi from UK. And the talk is on nano-optics. So please join back in 10 minutes.
No, it's one right.
Have to say it before other people.
Can I open?